Dude, this video is coming out on Friday, god dang it. That means I can't make videos on AP Chem or AP Physics or AP CS. God dang it. Literally my 3 favorite APs. That's so sad. Well, it turns out that AP Bio is on Monday, so, I mean, I'm not even taking AP Bio, god dang it. But hey, it's AP week, gotta do APs. Also, if you didn't notice, look at these beautiful cubes. I'm just gonna flex them from now on, you know what I'm saying? I mean, the main reason why they're still here is because I'm too lazy to bring them back to my room. So they're just gonna stay here. Get distracted too easily, god dang it. Bruh. Okay, we're gonna make this guy into something cool. Okay, it took me way too long to make a square. We're going for something cooler. Okay, that took way too long, bruh. Well, it's kind of cool still though. Oh my god, that is kind of hot. Not gonna lie. Okay, I'm done flexing. Okay, we're actually here to do work. Don't worry. We're doing work. We're doing work. Okay. Hello everybody, I'm Karar, and today we're going to be walking through the 2019 AP Biology free response questions. Let's do it. 1 hour and 30 minutes on the clock. Hopefully it doesn't take that long. 90 minutes in heaven? No, we are not spending 90 minutes in heaven. This is 90 minutes in auxin plant nonsense. Alright, auxins are plant hormones that coordinate several aspects of root growth and development. Indole 3-acetic acid is an auxin that is, okay, but boring. Circle one arrow that represents transcription on the template pathway. Identify the molecule that would be absent if YUC is non-functional. So transcription is converting a gene to an mRNA. So we could just circle this one over here. Why don't we just copy this whole thing into Photoshop? That seems like a good idea. Alright, epic, we get the nice low-res resolution, but anyways, basically transcription is whenever you convert a gene into an mRNA. So we take that. Okay. Identify the molecule that would be absent if enzyme yuck is non-functional. So it wouldn't get to here, right? Because all of these stuff will work, and then this guy is not working, so this arrow just doesn't make any sense. So this won't be produced. Predict how the deletion of one base pair in the fourth codon of the coding region of the gene TRPT would be most likely explain the production. Wait, what the heck? God dang it, I thought it was cool rapping, but it didn't work. Most likely affect the production of IAA. Okay, so basically in the TRPT, if you delete one, basically that's going to be a frameshift mutation. Because I'm assuming that the protein is like really big, right? Or at least not more than a lot more than 4 codons long. So if you mess up one base pair, it's like going to completely destroy the protein because it's going to mess up all the amino acids after that. So this guy's not going to work at all. So basically we could say it will prevent the synthesis. And could we just say since frameshift mutation? Sure. Dude, I haven't looked at an AP Bio rubric in forever, and I've also not studied for this, so that's kind of problematic, and I heard that the rubrics are really troll, so hopefully we say all the rubric stuff. Since frameshift mutation, and what else would probably be on the rubric? And then non-functioning protein to convert TRP to I3PA. Epic. Explain one feedback mechanism by which the cell could prevent production of too much IAA without limiting I3PA production. So basically, like, if you don't want to limit this production, right, what you got to do is you got to break up this enzyme and reduce the activity of that. So basically, there's a couple ways that you could limit production, right? You could either limit it at transcription when you're converting to mRNA, or you could limit it at translation, but generally, like, the most straightforward one is to just limit the transcription of the gene. Or saying a feedback mechanism, does that just mean that like IAA like inhibits that? Yeah, I think that works. So negative feedback, oh, I guess that's fine, of IAA on YUC. Or it could also be the transcription. What are we supposed to say? Are we just supposed to say? Yeah, I think that should be fine. Okay. Rhizobacteria is a group of bacteria that lives in nodules on plant roots. Rhizobacteria can produce IAA and blah blah. Okay. Plants release carbon dioxide into the node. Carbon-containing compounds. Okay, based on this information, identify the most likely ecological relationship. I mean, it's probably mutualism, right? Mutualism. Because both are being benefited, right? The plant gets nitrogen compounds from the bacteria, and then the plants give carbon-containing molecules to the rhizobacteria. Now, the second part's kind of sketch. Describe one advantage to the bacteria of producing IAA. So if it regulates like root growth and development. Well, yeah, why the heck would the bacteria produce IA? I don't get that. Maybe, well, I guess it could, oh, okay, I think it's probably like coordinate the growth of the root nodules, right? And then for E, a researcher removed a plant nodule and identified several cheater rhizobacteria that did not produce IA or fixed nitrogen. Describe the evolutionary advantages of being a bacterial cheater in a population composed of non-cheater bacteria. Well, basically, like, the other rhizobacteria are going to make all the IA you need, and then that will form the nodules, and then these cheater bacteria could just get all the carbon compounds from the plant without having to worry about producing all this nonsense. So the main advantage is that no work, but still gets carbon compounds, basically. And then the change in the population would mean that more of these bacteria become cheaters, in which case it would reduce the amount of carbon-containing molecules, right? Because if more of them are cheaters, there's going to be less nitrogen fixed. And then the plant will produce less carbon-containing molecules because there's less nitrogen fixed. Alrighty, let's check our answer. Is that it? Yeah, that's it. Alright, so you could either do this one or this one, makes sense. IAA, okay, good. Reduction, or, okay, that's basically what we said. And basically, yeah, you're gonna have a non-functional enzyme. Okay, epic. Negative feedback, okay, yeah, that's basically what I said. Nice. Mutualism, it lets them form the nodules, very cool. Oh, would you have to specifically say this? Okay, I guess in the actual thing you have to specifically say that they get carbon-carbon containing molecules, because yes, that is what they get paid for. Bruh, do you literally have to say, have more energy for reproduction? Okay, yes, sure. And then decrease in the, yeah, so basically decrease in non-cheater and increase in cheater, okay? And then that'll mean less nitrogen, makes sense. Okay. Alright, let's move on to the next one. Ooh, very nice growth curves. Okay, anyways. A student studying two different aquatic plant-eating unicellular what the heck am I on, bruh? Protist species A and B, are designed in an experiment, oh my god, designed in an experiment to investigate the ecological relationship. Okay, so group 1, they're grown in separate, 2, they're grown together. So the student placed 10 individuals in species A into a container. Okay. Alright, so, like, when they're separately grown, this guy, species B, grows pretty slowly, and species A grows like twice as fast, basically. Okay. But then once they're put together, like, species A still grows as fast, but then species B dies out. Growth curves in species B in group 1 and okay, have been plotted on the template. Use the template to complete an appropriately labeled line graph to associate, okay. What are the oh, very nice. Let's snip tool this boy in there. Alright, and then what do we got to do? So it basically wants us to add the other growth curve. So for group 1, species A, it starts at the same, but it ends at a much higher thing. And this over here is 1400, so it should go all the way to the top. Why is it so hard to draw? Okay, so this is going to be species A, group 1. And then the other one is species B, group 2 we want. Okay, so it goes up to 50. Where the heck is 50? So it's like tiny. Oh, bro, like literally down here. Hola mola. Bro. Okay, yeah. And then this is species B, group 2. Okay, I'm sorry I didn't plot all the points and nonsense. I promise, I would do that on the actual thing. Probably wouldn't, honestly. But anyways, we're moving on. I don't feel like graphing nonsense. As shown in the table, the student established treatment group 2 with 5 individuals of each species. Provide reasoning for the reduced initial population size. Reasoning for reduced initial population size? Why? Like, wouldn't it work perfectly fine? Or maybe it's like, if you have more, then resources will get used up faster or something? Oh yeah, I guess, larger populations would require more medium. Because you're comparing 10 people in the same amount of liquid, so if you put 10 of each, then you'd have 20 bacteria competing for the same amount as the 10 were originally in the group 1 stuff. So that makes sense, okay? And then for C, student claims that species A and B compete for the same food source and provide 2 pieces of evidence from the data that support the student's claim. So the first evidence, right, is like when they're grown separately, they both grow exponentially. Or, well, not really they grow logistically. When separate, they both grow to a certain carrying capacity. However, once they are together, species A grows to its carrying capacity, but then species B, it dies out because it no longer can compete with A for food. And then predict 2 factors that will most likely limit the population growth of species A in treatment group 1. So basically, the first factor is for sure going to be like your food availability. And then another thing that might impact its thing would probably be like the amount of space it has in the medium. Because bacteria do that thing, right, where when they're really dense, they can't really reproduce. Like quorum sensing or something? Yeah, something like that. Okay, E. Many protists contain an organelle called the contractile vacuole that pumps water out of the cell. The student repeated the experiment using growth medium with a lower solute concentration. Predict how the activity of the contractile vacuole will change under new experimental conditions. So basically this is a very interesting thing to talk about, because basically the way it works is that water wants to equalize its concentrations everywhere, right? So if you have like a lower concentration outside and a higher concentration inside the cell, then water wants to go into the cell so that it can dilute the cell more so that the concentrations could get equal. Now, of course, the cell doesn't want to get diluted. Why would you want to dilute cells? So that's why it has a contractile vacuole that prevents it from getting too dilute. So in the case where you have lower concentration outside of the cell, that means that more water is going to try to come into the cell. So that means that your contractile vacuole is going to have to be even more active because more water is going to come in. So you got to get more water out. So it's more active because more water entering. Alrighty. Is that it for number 2? Epic. Okay. Well, we had to label axes. I'm so bad at this game, bruh. Whoops. Well, how do you... What? Rip. Okay, so this is time, and then this is going to be population. Pretend that's like outside the graph. And then there's hours, and then like, yeah, that should be fine. Okay. And then do we just put like individuals here? Okay, and then for B, reduced initial population size, keep the total number of organisms the same in all containers. Okay, good. And then yeah, population density. Okay, so 1 point per row. So we got 1A to 2A. No, so we have, what do we have? Yeah, we did basically this one and this one. Okay, prediction, acceptable factors include food, space. Okay, we already got the first 2, epic. And then more active and yeah, more water is entering the cell. Okay. I think we're doing pretty okay. Let's get through this. Let's do the next one. Alright, pyruvate dehydrogenase complex catalyzes the conversion of pyruvate to acetyl-CoA, a substrate for the Krebs cycle. Okay, the rate of pyruvate conversion is greatly reduced in individuals with PDC deficiency, a rare disorder. So basically the way it works is that glycolysis happens, right? And then it like transports the pyruvate into the mitochondria, and then what happens is the Krebs cycle occurs. So that basically means that PDC should be more active in the mitochondrial matrix. Alright, A is mitochondrial matrix. So basically the mitochondrial matrix is the inside of the mitochondria, and then like the intermembrane space is what's in between the membranes, and then the outer part, like outside the mitochondria, is the cytosol. Okay, and then B, make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by the Krebs cycle. So it should not affect glycolysis at all, right? Because basically it's stopping pyruvate from being converted to acetyl-CoA. So basically PDC is converting pyruvate to acetyl-CoA, right? And pyruvate is a product of glycolysis, but it is the beginning part. You need acetyl-CoA in order to make this Krebs cycle happen, right? So without this enzyme, you're not going to have enough acetyl-CoA for the Krebs cycle to happen, so that's going to be reduced. However, glycolysis is not impacted, right? Because like this is after glycolysis happens, so it's not going to impact it at all. Now, the reason why I don't completely like this question is I'm pretty sure that there are some feedback loops that you got to consider when talking about this, because I'm pretty sure citrate or something exerts a negative feedback loop on glycolysis, so that if the citric acid cycle is not happening, then your glycolysis is not going to happen as fast. Okay, anyways, but that should be what they're looking for. PDC deficiency is caused by mutation in the PDHA1 gene, which is located on the X chromosome. A male with PDC deficiency and a homozygous female with no family history of PDC deficiency have a male offspring. So basically, if the female had no family history, then she's probably homozygous, the normal stuff, right? And then, I'm assuming it's recessive, right? Wait, how do I know if it's recessive or not? Oh, it doesn't even matter. Okay, anyways, yeah, it doesn't matter. So, it's X, so it's X deficient Y with X normal, or yeah, X normal, and you're gonna get, oh no, wait, there's like no probability, because basically if you get a boy, right, he has to get the Y from his dad, so this guy's going over there, and then the mom has to give one of her normal X's, and then her baby is going to for sure have XY, which is completely normal. You're not going to have a PDC deficiency. Epic. Okay, mitochondrial matrix, very cool. No change in glycolysis. Wait, I just want to search this up because, okay, yeah, okay, epic. But anyways, let me search this up. This should be citrate feedback loop. Oh, I love Khan Academy, but basically, I'm pretty sure it says it somewhere. Where is it? So basically, PFK is phosphofructokinase, and that's like a really important enzyme in the glycolysis pathway. So when citrate like builds up, that means that the citric acid cycle is not happening. So basically, technically, in our case, if the citric acid cycle is not happening, your glycolysis will also slow down. So technically, this answer is not completely correct, but I get what they're going for. And then our last one's right. Noise. Alright, acetylcholine is a neurotransmitter that can activate an action potential in a postsynaptic neuron. A researcher is investigating the effect of a particular neurotoxin that causes the amount of acetylcholine released from presynaptic neurons to increase. So, describe the immediate effect of neurotoxin on the number of action potentials. Okay. Predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. So the most important thing to know about action potentials is that the only thing that could change is how frequent they are. You can't change how much potential it changes. So if you add more, it's going to be more frequent, so more action potentials. However, you're not going to change the maximum potential that you reach because you can't change the amplitude of an action potential. So no change in maximum potential. And then the researcher proposes two models, A and B. Okay, so to prevent the effect of the neurotoxin, it's added to the synapse, and B, it's added to the cytoplasm of the postsynaptic cell. Wait, bruh. Big brain. Okay, basically, these neurotransmitters have nothing to do with the cytoplasm, okay? They're basically going to be released into the synapse, and then they're going to be recepted by stuff on the membrane. Nothing happens in the cytoplasm, okay? Especially not with acetylcholine, because that's not a, like, steroid hormone or anything. So basically, the model B is going to be useless, and then model A is going to be pretty useful because that's what acetylcholinesterase does. It breaks down acetylcholine. Very cool. Let's check our answers. It will increase the number, and it will stay the same. Very cool. Model A is effective, and Model B is not effective because it's not in the cytoplasm. Cool. Alright, number 5. Oh yeah, let's go. What is it called? Phylogeny. I love phylogeny. Don't you guys love worming out nematodes? Actually, wait, do you guys even know how to learn about nematodes and stuff in AB Bio? I'm pretty sure you don't. Well, I mean, if you're doing usable, you gotta know all that nonsense. It's so triggering. I love nematodes. Anyways, a researcher studying the evolutionary relationship among 5 primate species obtained data from a sequence of And then, based on fossil data, the researcher estimates that humans are the most closely. What the heck? Why are these so long? God dang it! Using this data, calculate the rate of empty DNA % divergence per millions of years between blah blah. Okay. So basically, your chimpanzees and humans are going to be the most similar. So we're going to do how many years ago did they diverge? 7 million years ago. So we got to take 8.8 and then divide it by 7 million or 7. And this is just going to give us, nice, I have my epic calculated right here. Dude, let me flex my Inspire too. God dang. Dude, it's actually pretty OP. This is way clunkier though. That's why I like my scientific. 8.8 over 7. 1.26, let's say. 1.26. And then, I don't feel like giving units because these are nasty units. 2 decimal places, cool. Using the data in the table, construct a cladogram on the template provided, provide reasoning for the placement of the gibbons on the outgroup of the cladogram. Okay, so basically gibbons have the most divergence, so like from humans, so obviously they're going to have to be the outgroup. Gibbons diverge the most from other groups. Okay, so we're basically going to do it in increasing similarity to humans. So orangutan is 16, gorilla is, let's see, where's gorilla? Gorilla is 16, 18, 10. Oh, this is so confusing. Why can't they just fill out the whole thing? So 16, 18, 17. 16, 18, 10. What's the last one? 10. So orangutan, then gorilla. And then chimpanzee is 8, 10, 17, 18. Basically, you're trying to go from lowest divergence to highest divergence. And then the next one would be chimps and then humans, I'm assuming. It doesn't really matter which order we put it in, though. Humans could go below chimps. Should be fine. Okay, and then C, on the cladogram, draw a circle around all the species that are descended from the species... What? Okay, so basically you just circled these 3 guys over here. Basically, you just look, like, where did it come from? So this one goes into there, this one goes into there, this one goes into there. So the top 3 gotta be from that circle, from that square. That it? Okay. Epicoo, even more epicoo. Leave similar. Okay. Noisu. 6, let's do it. The yeast Saccharomyces cerevisiae is... Why are these questions so long, bro? Occurs through metabolic path. Okay, Jesus. Identify the role of treatment 1. Yeah, I just don't want to read this. In the experiment. All amino acids are present. That's basically going to be a control, right? What are you even trying to do? Yeah, I think that should be a control. And it should be a positive control. Is that what it wants? Positive control. How about that? Basically, the difference between positive and negative, I learned this recently. Positive means that you give treatment 1 something that you're not giving to your experimental group. And like your experimental group, and if it's negative control, then you're not giving your like treatment 1 the treatment that you're giving to your other groups. Provide reasoning to explain how mutant 1 can grow on treatment 1 medium but cannot grow on treatment 3. Basically that means that it needs methionine, right? Because if it could grow on 1 but it can't grow when it's missing methionine, that means it needs methionine to survive. It cannot synthesize it on its own from other ones. Okay, and then C, yeast mate by fusing 2 haploid cells to make a diploid cell. In the second experiment, mates the mutant 1 and mutant 2 to produce diploid cells. Using the table provided, predict whether the diploid cell will grow on each of the 4 mediums. So basically, if you're making 2 haploid into a diploid, they retain all the DNA from all the other ones, right? So essentially, mutant 1 will have the ability to synthesize, let's see. So mutant 1 is able to synthesize leucine, and mutant 2 is able to synthesize methionine. So overall, they should be able to synthesize everything because they combine their genes. However, if no amino acids are present, then that might be a problem, right? No amino acids present. Why is the wild type able to survive on that? How did that even work? Oh, they only have single recessive mutation. So I'm assuming that the reason why mutant 1 can't grow when no aminos are present is because it can't synthesize methionine. So I guess we'll just go with plus for all of them. It should be plus for all of them. Okay. Well, yeah, because they have 2 separate recessive mutations, right? So if you combine them, they'll have at least one of the normal dominant one, and we should be Gucci. What the shoot? 5 pluses. Let's go. Okay. A positive control. I don't know. I can't tell if these bullet points mean that you have to have all of them, or whether. Well, anyways, this is just stuff that you gotta add, but once you get this, you're just basically just listing the definition of a control. Okay. Can use methionine when present, but it cannot synthesize it. Okay. There will be a growth in all 4. Epic. A researcher is studying patterns of gene expression in mice. A researcher collected samples from 6 different tissues in a healthy mouse and measured the amount of mRNA from the 6 genes. The data are shown in Figure 1. Based on the data provided, identify the gene that is most likely to encode a protein that is essential for I can't speak this fast. Why am I so bad at this game? Essential component of glycolysis. Okay. Provide your reasoning to support your identification. Okay. So your glycolysis is going to be very important in your skeletal muscles. So should be one of the genes in muscles. So it's either gene G or gene I, but oh wait, yeah, but your brain is for sure gonna have a lot of this glycolysis happening because your muscles and your brain use up most of your glucose. So gene G. Wait, bro, they literally made the answer the one that starts with the same letter. So gene G, since brain and muscle need a lot of it. Muchísimo. Okay, B. The researcher observed that tissues with a high level of gene H mRNA do not always have gene H protein. What does having high mRNA levels have to do with protein? I mean, you could have a lot of that, but it just would not produce protein. Could we just say post-translational modifications or something? What else would contribute? You could also degrade the mRNA, right? You could also break down the proteins. Yeah, I guess you could just say post-translational modifications. I'm pretty sure post-translational modifications is what it's looking for. And you could be more specific. You could say that maybe the RNA is degraded before it could be turned into a protein, or that they have a certain molecule that prevents the ribosome from binding, something like that. Okay, gene G is the only one that's expressed in all 6. Okay, yeah, that's true. Oops. Bruh. That would make a lot more sense. But I mean, my explanation is a lot valider, just gonna say. Hey, we got it, let's go. Very cool. Alright, last question. The petal color of the Mexican morning glory changes from red to blue, and the petal cells swell during flower opening. The pigment heavenly blue anthocyanin is found in the vacuole of the petal cells. Petal color is determined by the pH of the vacuole. Okay. Identify the cellular component in the model that is responsible for the increase of pH of the vacuole during flower opening. So is it just gonna be proton pump? Yeah, proton pump. Oh, increase in pH. What the heck? Oops. K plus H plus transport protein. Since it removes H plus ions from the vacuole. What does it mean, the component's role in changing the pH of the vacuole? Is that just what it wants? I think that's all I could say. A researcher claims that the activation of K plus H plus transport protein causes the vacuole to swallow with water. And that makes sense, right? Because you're basically pumping in solutes, which are K plus ions, right? So you're increasing the concentration of K plus, but the K plus cannot escape. So the membrane is like impermeable to K plus, right? So now, in order to equalize the concentration, the K plus can't come out, but the water has to go in. So that makes sense. K plus can't escape, so vacuole becomes hypertonic relative to the cytoplasm. Okay. Hypertonic, hyperosmotic. Okay, very cool. We made it through. I hope that was helpful. I don't know whether it was, but it's basically the last MAP that I can cover, so I'll just put out a video there. So, hope this helped. If you guys want any specific videos, just let me know. APs are basically over. I have like 2 APs next week, but one's Spanish, which is troll, and stats, which is also troll, so that's fine. Alright, thank you guys for watching so much. As always, if you enjoyed the video, leave a like and subscribe for more. Thank you guys for watching again. See you guys next time.